Well, thanks very much to Rivka and the organising committee for inviting me. NMR is very close to my heart. I have been doing NMR, not so much recently, but a lot since my undergraduate days and PhD. And Glasgow is also very close to my heart, and I haven't been here for a long time. So thank you again for inviting me. Today, I'm going to talk about work from a PhD student in the lab, Harry King. He was joint student with Kat Artavanis-Sakanas, who's in the Department of Pathology at Cambridge. And this was a collaboration with her group and also with Hiro Suga's lab in Tokyo. All the NMR was carried out by Mark Bycroft, a collaborator in Cambridge, and performed at the Crick Institute. And so we're very grateful for the NMR time we received there, as well as the support. So the protein I'll be talking about today is involved in malaria. And this is obviously a devastating disease. It's caused by the Plasmodium parasite. It's found in countries all around the world and is responsible for over 500,000 deaths. And the problem is that there's no vaccine currently, and treatments are increasingly becoming ineffective because of drug resistance. So Kat's lab studies the ubiquitin proteasome system in Plasmodium. And this is a particularly interesting topic because of the parasite's complexity. So this parasite can replicate both sexually and asexually. It can infect multiple hosts, a vertebrate and a mosquito, and it can infect multiple cell types, including hepatocytes and red blood cells. And it does all of this with a genome that's smaller than the size of yeast. So by necessity, post-translational modifications must be playing an important role in its development. And specifically, the ubiquitin proteasome pathway is at the core of Plasmodium life cycle transitions. And this is for two reasons. Firstly, they undergo some huge morphological changes shown here. And they also undergo multiple rounds of rapid replication shown here with the arrows. And both of these are heavily dependent on protein turnover, and that is, of course, underpinned by the ubiquitin proteasome system. It's a pathway that's essential in Plasmodium and, in fact, in all eukaryotic organisms, and it's highly conserved as a result. And this has a number of benefits, but also presents challenges when it comes to drug discovery. So the ubiquitin proteasome system is summarized here briefly. It requires 3 enzymes, an E1 activating enzyme, an E2 ubiquitin conjugating enzyme, and an E3 ubiquitin ligase that together work to attach a polyubiquitin chain onto a target protein. And importantly, this polyubiquitin chain conjugation is reversible, so that the chain can be removed by deubiquitinating enzymes or DUBs. And one of these is the focus of the talk today. It's a DUB called UCHL3, and it was identified in Plasmodium falciparum by Kat's group a number of years ago. They determined the crystal structure of the protein on its own, shown in blue here, and in complex with ubiquitin, shown in gray here. Ubiquitin is in green. It has very similar structural and functional features to the human enzyme, shown here in orange, and it's essential to parasite survival. It's expressed across the whole life cycle of Plasmodium, both in the human and mosquito stages. And so consequently, it must have a very fundamental role. And this makes it an interesting drug target. So we set out to develop inhibitors against this protein. And the approach that we used was constrained peptides. So this involves locking a peptide in its bioactive binding competent conformation. And this can be done in a variety of ways, either by cyclizing the peptide or indeed making bicycle peptides, and also by stapling the peptide. So this is using chemically chemical cross-linkers. And as well as being used to enhance the peptide's affinity for its target, peptide stapling has a number of other advantages. It increases the peptide's resistance to proteolysis, and this is, of course, very important if it's going to be used as a drug. And it can make peptides cell permeable, and that's very important where you have an intracellular target, of which UCHL3 is an example. And a number of these constrained peptides are in clinical trials or in the clinic currently. We employed an mRNA display system called RAPID. This stands for Random Non-Standard Peptide Integrated Discovery. It was developed by Suga's group in Japan, and he spun out a very successful biotech company from it called Peptidreams. And these libraries are composed of around 10 to the 12 cyclic peptides, each between 6 and 15 amino acids. They importantly can incorporate natural amino acids, and that gives them much greater chemical diversity. It also increases other properties such as resistance to proteases and cell permeability. So we screened these libraries against the Plasmodium UCHL3 protein. Importantly, we want to inhibit the Plasmodium protein, but we don't obviously want to inhibit the human protein. And remarkably, Hiro reassured us that we wouldn't need to do a negative screen against the human protein. And this was very surprising because the two proteins are very similar in sequence. But he told us that these screens are so selective that we wouldn't really have a problem. So we screened these libraries. We immobilized the UCHL3 with streptavidin beads. And after 7 rounds of selection, we obtained a library of 10 peptide families. The top 10 of these sequences are shown here. These are all scrambled. They're all cyclized, and they range in length, as you can see, from 12 to 17 amino acids. And at this point, we were going to do more screens. We were going to look at shorter amino acid, shorter numbers of amino acids in the peptides. Also look at incorporating more unnatural amino acids. Here we only have an unnatural amino acid at the N-terminus that allows us to spontaneously form cyclic peptides with a cysteine at the C-terminus. But Harry was in Japan at the time, and then the pandemic hit. So he came back home just in time, and we looked at these top peptides that we had from this screen. We used bilayer interferometry, or BLI, to look at the binding affinities. This is a surface technique similar to SPR or Biocore. We have biotinylated Plasmodium protein immobilized on the sensor, coated with streptavidin, and then flow over the peptide shown in yellow. And as you can see, all but one of the peptides gave us good data with KDs in the low nanomolar range. So that was very pleasing. You can see that some of the data here, very nice high affinities, very slow off rates. As I said, the BLI data look very convincing, but results for an orthogonal assay that we wanted to use, a thermal shift assay, these were somewhat contradictory. So this assay is shown here. We use a dye called Cipro Orange, and this is a fluorogenic dye. When it binds to hydrophobic residues, when the protein unfolds, the fluorescence increases. And so it can be used to measure the melting temperature and ligand binding, which should cause an increase in melting temperature. And one of these TSA assays is shown for one of the peptides. At increasing peptide concentrations, you can see that the melting temperature increases. In this table, we've listed all the changes in melting temperature for all the different peptides at one peptide concentration. And you can see actually for only 2 of them do we see a significant increase in melting temperature. All the other peptides have very small changes. Some of them have decreases in melting temperature. So we've looked at binding. Binding doesn't necessarily mean that we have a peptide that can inhibit the enzyme. We wanted to test inhibition of the DUB, and we use an assay where we incubate UCHL3 with a fluorogenic ubiquitin substrate, UBAMC. This is shown here. So this is conjugated to the C-terminus of ubiquitin, and when the DUB binds and cleaves off the AMC, it becomes fluorescent. So we can measure the increase of fluorescence as the DUB cleaves the ubiquitin as a function of time. So this is the assay again in the corner here. We had a positive control, which was the enzyme with the substrate. And that's shown here. We had negative controls of no substrate, no enzyme, and also in green here at the bottom, enzyme with something called NEM, which is a suicide inhibitor. This alkylates the active site cysteine of the enzyme. So as you can see here, the main results were that some of these peptides, including the one shown here, showed a really nice dose-dependent inhibition. And these peptides inhibit even at nanomolar concentrations. And most importantly, they are exceptionally selective for the Plasmodium protein. So this is the human UCHL3. You can see even at high peptide concentrations, we don't get any inhibition. And that is really striking because the human and Plasmodium protein have very high sequence identity. So in red are the active site residues, and in highlight are the residues that bind to ubiquitin. And you can see overall very high sequence identity. So we've pulled out peptides here that bind selectively to the Plasmodium protein, but not to the human protein. Oh, and I should also say that ubiquitin itself in Plasmodium and human differs in only one amino acid, so that the enzyme substrate interface is also very highly conserved. We wanted to determine the structure of the complex of UCHL3 with the two best peptide inhibitors, so we could identify where the binding site was and how they inhibit. We first tried crystallography. We got very nice crystals, but none of them contained the peptide. So then we moved to NMR. And as I said, all the experiments were performed at the CRIC by Mark Bycroft, a collaborator in Cambridge. We first needed to assign the spectrum of the UCHL3. This was reasonably straightforward. It's a very well-behaved protein, very soluble, expresses in very high yields, and it's 27 kilodaltons in size. So when we incubated the protein with the first 6 of the peptides, we didn't see any binding. And these were the peptides that appeared to bind by BLI, but didn't inhibit the enzyme. The 2 best inhibitors were the last to be tested, and these did bind, which was a big relief. We were worrying that we'd wasted our colleagues' time at the CRIC. And these there was slow exchange between the bound and the unbound forms of the protein, and very large chemical shift changes. So the spectrum had to be reassigned for each peptide. The fact that the slow exchange confirms that there's high affinity that we saw by BLI. And because these are relatively large peptides, they have a number of aromatics in them. That's why we see these very large chemical shift changes. We next looked at identifying the binding sites. So these are peptide interaction heat maps, colored according to the size of the chemical shift changes from blue to red for peptides 60296 and 60297. The largest chemical shift change is mapped to the ubiquitin substrate binding site on UCHL3. And this peptide in particular seems to cause quite extensive remodeling of the whole of the beta sheet, which is kind of adjacent to the binding site, and appears to expand the binding interface relative to that of ubiquitin. As I said, the biggest change is mapped to the ubiquitin binding site. You can see from the structure here that there's very close overlap. between where ubiquitin binds, ubiquitin is shown in yellow here, and these the chemical shift changes on the protein with the peptide bound. We also looked at simulations. This is in collaboration with colleagues at Queensland University. They used the heat maps as constraints, and you can see that the pose of ubiquitin and the peptides is very similar. The interfaces overlap really very well. And cyclization of the peptide appears to lock them in binding competent conformation, which is the whole purpose of cyclization of peptide constraint. So to summarize, we were able to use a rapid screen against Plasmodium UCHL3 to identify cyclic peptides with low nanomolar affinity for the protein. 4 of these peptides selectively inhibited the Plasmodium protein versus the human protein. NMR allowed us to identify the binding site on the peptide, on the protein, and we could see that inhibition occurs because the peptides block ubiquitin binding site. Importantly, also NMR identified several peptides as false positives from the screen and also from the BLI. We suspect BLI, because it's a surface technique, was the reason why we got some false positives. So going forward, as I said, we had hoped to do some more screens to incorporate numerous other unnatural amino acids to make the peptides shorter. And this would help us to get these peptides into cells, and then we'd be able to test their effects in vivo. This is quite a challenge. So peptides always have to be cell permeable, but in the case of malaria, there's a number of membranes to cross. So the peptides have to cross the membrane of the cell it's infecting, the parasite vacuole that's membrane that's there inside the cell to protect it, and then the parasite cell membrane. So this is a big challenge, but we hope with using the use of unnatural amino acids, it might be possible. Before I finish, I would like to just briefly mention another project that we've very recently been carrying out. And this really highlights how important NMR is in a biophysicist's arsenal. So this is a project on this protein in green here, PR65. It's a heat repeat scaffold subunit of a heterotrimeric protein phosphatase, PP2A. And you can see it's in complex with the catalytic subunit and the regulatory subunit here. It's been proposed to act as an elastic adapter. So these repeat proteins are elastic, and the idea is that it brings the substrate, which is bound to the regulatory subunit, into contact with the catalytic subunit, allows dephosphorylation to occur, then opens up and releases the substrate. And for multiply phosphorylated substrate, this open and release and closing again allows processive dephosphorylation to occur. A number of small molecules have been identified. These are small molecule activators of PP2A SMAP. There's some data in cell-based phenotypic assays that they're activators, but the mechanism of action is completely unclear. And we've been using a range of techniques, including single molecule optical tweezers, to look at the force response of PR65. As I said, it's an elastic repeat protein. It opens and closes in a reversible manner. And this is the protein without SMAP. So we see a number of force peaks, which correspond to individual repeats unfolding. We can pull on these proteins and release the force multiple times, so they can go through multiple stretch, relax cycles without the protein, without any hysteresis and without the protein irreversibly unfolding. And so with the SMAP shown here on the right, we saw some really dramatic and also very long range effects on the protein, including very large increases in mechanical stability. So you can see the forces here are in the tens of piconewtons, whereas without the SMAP bound, the forces are sub 10 piconewtons. And because these effects were very striking and quite unusual, we wanted to confirm binding by a more conventional and ensemble method. And we worked our way through a really exhaustive list of techniques, fluorescence polarization, circular dichroism, without any decent results, mainly because these small molecules are really insoluble. So at the concentrations we needed, we know that this, we think that it has a micromolar affinity. We just couldn't get high enough concentrations of SMAP. So we encountered the same problem with all of these techniques. And then finally, we approached Daniel Nietlisbach, our neighbor in the biochemistry department, and asked him if he would do some NMR on the small molecules. So he performed STD and fluorine NMR. He also performed water logsy, but again, we had the same problems where we had very low solubility. But finally, with this NMR technique, we were able to confirm binding. So yes, to summarize, NMR is a really important technique. In this project, it allowed us to confirm that these small molecules were binding and to confirm these very unusual single molecule effects. And in the previous project, it allowed us to map the binding site of the peptides on the protein and also to identify peptides that were false positives. So with that, I'd like to thank you for listening. I'm happy to take questions.